honor to introduce you Dr. Rebecca Todd. Her work has been in understanding the connection related to emotion and health and pathology. Rebecca, we have a common interest. I don't know if you're going to guess, but so Rebecca is a dancer and a choreographer, or was?
Used to be.
Yeah. And I was also a dancer. So,
I didn't know that. That's
yes,
awesome.
yeah.
We'll have to bond over that.
Yes, during coffee break. So that's really great. I'm very impatient to hear your talk. And so I give you the stage.
Thank you. So yeah, as Sylvain said, my lab works on our basic research. I'm in psychology at UBC, and our basic research is on interactions between cognition and emotion in non-clinical samples of adults. And then we do a lot of collaborative work with folks both in behavioral neuroscience and in psychiatry, trying to do translational work, bridging, what we know from rodent research about the circuitry and pharmacology underlying certain patterns of behavior that then we hope will predict certain subtypes of, say, depression in the clinic. But thanks to Fidel, who left, but I felt like he really got the assignment of these ideas talks. And that was a great discussion. And I'm going to be more didactic because I want to lay out some information about a methodological approach before opening it up to discussion and questions again. But just my personal, Fidel talked about how during COVID, he discovered complexity theory. So I was sort of raised, steeped in complexity theory. But in my work as a cognitive neuroscientist, I've really been working with this tension where my understanding of the brain is of a complex adaptive system embedded in a complex adaptive system embedded in a complex adaptive system. At the same time, the methods I've been trained in and use and have gotten traction out of to publish with are linear all the way down. And so I've been living with this tension for a long time where I have this concept of how things really are that is not reflected in the methods I have at my disposal. And so this idea of the person and the brain as complex adaptive embedded systems has remained metaphorical and a little bit isolated from the actual empirical work that I've been doing. And part of that is because, so Sam gave a rundown yesterday of a long history of work that I've been aware of and admired, but I don't have a background in physics or engineering. And so I didn't feel like a really solid grasp of the tools to look at the brain and the person and the environment as complex adaptive systems were within my disposal. I think that may be changing now, which is why I'm excited. So I'm now going to switch gears a little bit. Let me see if I can make this go. Yeah. So what I'm actually going to be doing is talking about a methodological approach my lab has been developing that's been really useful and that I'm very proud of, but so far it's rather static and it's linear models all the way down, linear models upon linear models. So I just want to sort of open up questions about reconciling it with some of these approaches to the brain as a dynamic system. So Brandon asked a question of Sam yesterday. And really what I'm going to try to do over the next 15 minutes is explain what Brandon was referring to in his question so we can again ask it of Sam and everybody else in the room, because it's exactly where I was sort of wanting to go with this. So I'm going to be talking about the work of James Crickleby, who was up until last week a postdoc in my lab. And I'm going to give you a big picture view of it. If you want some details, Brandon Forrest here has been working in the trenches on it, and he can answer some of the details. But really, so what this is we've been using this multivariate approach to sort of decoding brain activity called representational similarity analysis, which many of you are probably familiar with, but I'm going to explain a little in a minute to get us all on the same page. With this approach called pattern component modeling, where you try to model the informational content that's being instantiated in the patterns of voxel activity across a certain brain region. And I'm going to unpack this. And I'm going to talk about what we've done with it so far and some of the traction we've got out of it, but also where we're sort of stuck in a static snapshot model. So how can we sort of integrate more of the dynamicism that we know is central to brain processes into it? Number one, can we? Is it useful to? And yeah, and is this even? Does it even make sense, basically? So let's start with just a little review of representational similarity analysis, or RSA. So that was introduced to the world by Nico Kriegskort in 2008, I think, with this paper here. And basically, what it allows you to do is you choose, in the sort of standard form, choose a region of interest in the brain, run a general linear model for conditions or stimuli of interest. In this example, it's houses and faces, individual houses and individual faces. Extract a beta value for each voxel. So each of these little boxes is just a example voxel from a region of interest, and look at the pattern of larger versus smaller beta values, basically, for each of the stimuli or conditions you're interested in. And what you can then do is create a correlation matrix. This is a dissimilarity matrix, but you can invert it and have a similarity matrix, which just is the degree to which the voxel patterns between stimuli or conditions of interest are correlated. So you go from voxel patterns to a correlation matrix, and we've been looking at a lot of correlation matrices yesterday. They were mostly correlation matrices of correlations between time series, between voxels or electrodes. This isn't correlations between time series, between pairs of voxels and electrodes. This is correlations of voxel patterns over at a spatial scale, over a region of interest, in response to different stimuli, okay? So what that gives you is you have a measure by the strength of these correlations of how any region of the brain is treating one thing as like or unlike another set of things. And the way that gets, you can then sort of put it into representational space, where you calculate a Euclidean or other kind of distance between the stimuli, and you can see sort of how close or distant the brain's response to stimuli is in representational space. And you can, of course, then look at that in different brain regions. In this case, they use regions in the ventral visual stream to see how different large-scale brain regions are representing these things as similar or different. So that's standard RSA. It's been in use for a long time, and it's been used by a lot of people because it's quite tractable, and you don't need any machine learning. You can sort of extract these patterns with regressions all the way down. I'm just going to show now a little example of, so we use this as a basis for sort of this pattern component modeling, to model what content each of these brain regions may be representing in a given situation. So I'm going to start with just the simple RSA part. So this is a very simple Pavlovian conditioning experiment where people learn to associate individual faces with either a soft brush or painful pressure. So as painful or pleasurable touch. Here's the voxels. Here's the bold time series in response to the voxels. And so here you have, we were interested in events associated with the experience of pressure pain, of pleasurable brush stroke. And then we wanted to see if these could predict the patterns that were read up by the brain when people saw the faces that were associated with these things before, during, and after they learned the association. Okay, so basic RSA, pretty straightforward. So this is where we bring in the pattern component modeling factor. So this idea, I think, was originally suggested by Nico Kriegskort in 2013, and his group has been working on it, but so have a couple other labs, including us, in parallel. And that is basically where you create a correlation matrix that models what the sort of ideal representations of specific content. So some of these methods that have come out are very data-driven, but Jamie's method is very hypothesis-driven. These are pre-specified models of the information that a given brain region may be representing. So in this case, a matrix, an ideal sort of pattern of interest, we call them, that if the brain was just representing painful touch, if it was just representing pleasurable touch, and then this is if it's actually representing touch, painful and pleasurable touch on a valence spectrum from good to bad, from painful to pleasurable. So these are just like examples of information content you can model. And then the idea is to test these models to see if you can predict the actual patterns, RSA patterns that we see in the brain. So just to walk through, these animations are all Jamie's, by the way. So you have your hand, which is being touched. And you might want to look at patterns in S1 that might reflect, say, discriminative aspects of touch. And you also might want to examine patterns in the ventromedial PFC, which you might expect represent more affective aspects of touch. You have your correlation matrices for each of these. So you take trials in which pain was experienced and correlate them with the other trials in which pain was experienced. You then throw the soft brush trials into the matrix, as well as trials in which the hand experienced no external touch at all. Okay, so that's your RSA. That's what your data looks like. You then test your pattern component models. So this is where we have really layers of regressions on regressions on regressions. So what Jamie then next does is he does a greedy best first search algorithm using regressions with Bayesian information criterion to test models of the combinations of these ideal patterns that best explain the data. So this is an example of a couple of patterns that best explain the data in these two brain regions. And this is the pattern component modeling. So you have for the VMPFC pressure pain, pleasant brush. And then for S1, you have other things that are more related to touch discrimination. And then in Jamie's wonderful animation, they slide together and you see how together they do a pretty good job of recreating the pattern of data that you actually observed. And then what he can do further is do a further round of regression and some cross-validation to see which of these ideal patterns is contributing most to the variance in any one of these regions. Okay, so this has been really useful. We've used it in a number of studies to be able to kind of look at questions of like, well, which parts of the brain represent discriminatory versus affective components of touch, and then moreover, with conditioning, what information do these regions of the brain represent before, during, and after learning? But again, we're getting these very static snapshots of the brain. We're not getting brain dynamics. So this is another sort of instantiation of the approach that Brandon has been working on with Jamie, and he can explain more of the details of this. But basically, this is from the same experiment, which was a collaboration with Adam Anderson at Cornell University, where we took some faces, novel faces, and slowly morphed them into faces that have been associated with pain or pleasure or neither. And then they create these sliding windows where as the face gradually morphs, they sort of slide over independent GLMs and create matrices, correlation matrices out of those. Again, Brandon can dig into the details of this approach, but we're starting to integrate time now. And so you can sort of take a face morph, you can predict the representations. For example, it might, some parts of the brain might represent, the continuous morphing that is the perceptual change that you're seeing, whereas other might actually sort of flip. At one point, they're perceiving one face, one identity, and then flip it into a new identity And that might or might not be different with conditioning, right? So you can take your predicted representations and show the observed data. So now we start to get a little bit more of a temporal element into it. But Jamie has really always wanted to actually do a sort of take this the next sort of next step dynamically. So far, we've been able to create these snapshots of information each brain system or brain region is modeling independently. But we've also observed that some regions represent fairly isolated bits of information, pain, pleasure, for example. Anterior cingulate seems to be dominated by representations of pain, the insula of pleasure. But then as you move to the amygdala, so misunderstood as a primitive brain region, you actually have an integration of more complex information where you actually see it representing valence on a spectrum. So one thing you might say is like, well, over time, like how do these representations propagate through the cortex? For pain and pleasure, it's kind of interesting because you actually have valenced receptors. You have painful touch receptors and pleasurable touch receptors. So you can expect them to propagate, representations of pain and pleasure to propagate independently through some brain systems, but other brain systems, you might have more complex sort of representation of information from the start. So this is all stuff that if we can start looking at this content instantiation, this information instantiation more dynamically, more consistently with what we know to be a sort of a dynamic system, then it might give us some traction, be a little bit closer to our understanding of how the brain works. Because again, we've been working with this tension where we have these linear methods that are useful methods, but that's not how the brain works. So my questions that I sort of want to bring to the group is, and this is really what Brandon was asking, is it useful and feasible to try to track how the information instantiated in these regions of interest as it propagates throughout the brain? Is the use of sliding windows a useful and appropriate approach? What are other approaches? Again, can we track the evolution of this at the rate of a TR? And then are approaches borrowed from dynamical systems theory appropriate here? You know, do we haul out our differential equations? Does it make sense, or is it just apples and oranges? And it's a different questions that we're answering with different approaches to it. So those are the questions I want to leave you with. I want to thank, these are the 4 people who have been involved in the study, and Brandon also makes a mean garden bed. Or answers. Do you want to take?
Understanding of how those pleasurable and painful stimuli propagate in the brain and get consciously realized as those sensations, which is, I know, what you're really interested in.
So we should be talking more.
Yeah, we will. But what I think we need to do, Victor talked a little bit about yesterday about moving from into a genetic. That's a whole different way of thinking about it. And if you use the idea of manifolds, then you can trace the trajectories on these manifolds, which will get there, which will get to what you're wanting to do.
And
now,
I
what's
think
the best?
My gap is, how do we do that? Sounds great.
The right metric words.
Where's Victor?
That's frustrating.
It's like right when the here. Sorry.
So I think use that kind of an approach, and maybe thinking a little bit about what Olaf said, yesterday about changing the way we're thinking about that, you know, it'll be a little bit of trial and error, but I think between those two concepts, we can come up with something that will work to answer the questions for particularly.
Yeah, I want that to be the next phase of my career in cognitive neuroscience, and hopefully Brandon's on board because he's the know-how in our lab right
No, I think that's a fantastic idea. We'll build on those ideas to.
We have an answer for how to measure a manifold.
Sure. I mean, there's a number of ways. The easy one, this is one everybody does anyways, some sort of principal components now of these data. The nice thing about what you've got here is you're integrating the brain and the cognitive competence. That's what's missing in a lot of the stuff that Vic and I have done, where we create a manifold of the brain dynamics, but they're sort of independent of the perception, if you will, or experience. What you've got here is the section of things. In some respects, you've got same manifold all the way down. It's basically what you've got is really looking at, you can create a manifold of brain dynamics. You create a manifold of the behavior dynamics, as long as the behavior has a temporal evolution to it. You've got here is actually a way to put those two together. And that's a simple expression of doing dimensionality reduction, allowing the base to evolve through the orthogonal projection, again, what PCA does, or you can think about it as non-orthogonal, depending on which fits better. And that's more of an empirical question, sort of analyzing this data. The nice thing is you can start tracing out the trajectories at an individual level as well. So if you have the space sort of constructed where you've got the general space, and then see how the individual trajectories transpire within that. You've got the static part is the stimulant, and that's the same for everybody. So that's sort of where you start from, and then you can then project back and see where they start diverging. And those are the critical individual differences.
But how do you quantify the individual differences?
The deviation from that sort of stimulus trajectory. You'll get a distribution across everybody, and then start tracing out the distributions where they start diverging.
One thing that's nice about this data set too is we have quite a few participants. Like we have nearly 100. So
And trial by trial.
and trial by trial, yeah, these are slow event related designs. So
Just one comment on that. I think like you point out is very important. I think there is like another component also that come back to your question, like and especially in this domain. I feel like the subtyping or trying to find ways to get at the individual level and is probably the first step into this type of like thinking dynamic. It's really important to kind of like not forget about that step.
Yeah.
Because whatever number of people we have, the issue is like we tend to kind of average, right?
Yeah.
And so as soon as we average, we kind of going on the opposite way of the difficult system. So this is like probably one of the two steps, right?
we actually we have a small amount of like genetic data on these participants, and we were sort of lumping them in conditioners and non-conditioners for a while, and it's at a certain point just didn't have enough data to really pursue that. But
There is one other aspect, it's kind of stupid you mentioned this, but you have virtual brain as another way of straining that space. And then you add the individual that got that intersection between the static stimulus representation, not based static brain, but the structural component of that you fixed as well. And you add on the individual dynamics and then start looking at deviations from that.
But couldn't there be two people with the same deviation but totally different trajectories?
Yes. Yeah.
Seems like a problem.
No, that's actually a part. That's the first thing.
That's a typical case where the dynamic system can really perform.
What you want to find out is when you start, I mean, it's going to be difficult with one data set to do that, but you start pushing the boundaries of how that person is behaving. And there's going to be a consequence for how their particular consequences could be bad or good consequence, but how they explore that space of possible trajectories. So if they have, say, 20 trajectories to get between A and B, and of those 20, 15 of those stay in the same part of the manifold. It could be the case that when you change the behavior responses, they can't adapt as well as someone else who actually.
Okay, I was thinking more about individual differences. If you have one number to compare people and two people have the same deviation, but totally different trajectories.
Yeah, we wouldn't use one number. No. We want to get away from reaction time. Yeah, that's about right.
I didn't say reaction time.
Well, that's an example, right? Reaction time's a point test.
Okay, really, that's a really cool discussion. Thank you, guys. I hope like all the students are growing. If you don't, just ask questions, please, like sometimes.
And I think we're recording, right? Because I plan to come back and
Could also make.
watch it.
I'm not sure if anybody on the Zoom audience has.
Oh, it's okay, that's a reader. Okay, I will check on the one. Sorry, there. No, we don't. Okay, so we're good to go for now. Okay, so wait, I think okay, let's go with Abdul. I'm trying to get the.
Okay, thank you for the presentation. I'd like to come to the notion of similarity. So if you have different similarity measures, it can capture different ways of the information, right? So if we, for instance, in the context of dynamical systems, so how do you take this into account? So based on the hypothesis that you are doing on the similarity measure, how is it taken into account? So for instance, if in the case of manifold, for instance, if we use Euclidean distance, this can be maybe more sensible to spherical shapes of the manifold. If we take another measure, similarity measure, this can capture another shape. So how do you take this into account?
Yeah, I haven't, like, I'm just starting to think this through right now. So my answer is, I don't know, but it's important to consider, I think.
Maybe anything you have?
The problem with any of these, it's a great idea. The problem is how to get enough data, individual to put it into that kind of representation. That's where I think we're having these intersection between something like virtual brain and empirical data. And then you use that virtual brain to generate these trajectories from individual, and you can start constructing that space.
We've been thinking, we've been talking, we've been starting to explore the virtual brain in our lab, just initial like download, but really wondering like how exactly that, how can it be useful in some questions that we want to look at.
And then the question then becomes, this is something we're going to explore together is what aspects of what's in the model are really representing the important individual differences. A lot of the stuff we're doing in humans, at least, is okay, but it's rather coarse compared to what we really need to do. If you compare, I think both Victor and I mentioned the mouse example where we used to do TBV mouse, virtual brain mouse. There you actually, because the resolution is higher, you do see that you capture individual differences quite well. In the human application, it's not as, we're measuring very coarse. So the DTI data we get from there is crap in terms of getting individual differences. Well, optical stuff is pretty good. We're still missing some of the stuff on the structural side, which we can improve. Aaron's not around, but we can probably get better on that domain to capture more of that variability.
And flexibility at the mathematical level, that's what you want to look for. In that case, any I see the questions.
First, I just wanted to say I didn't know you're representationalist. Just use the word representational.
I know, it's divorced for having that title. Instantiation, but then, in the heat of the moment, I say representation is a flaw. My parent who wanted to divorce me when he saw on Twitter that I had representation.
I'm just trying to look out for you.
No, I'm already in trouble.
So, yeah, I think we talked about this before, right? But it talk stimulated again for me, which is the idea of not only, I'm basically talking clean as voodoo dolling me right now. It's like, it's not it's not only content, right, but it's the manner of change that I guess this dynamic RSA affords you, right? You can ask that question about how things are changing, obviously, which I think it's inevitably related to the content, but gives you a whole new variable to again draw associations with other variables. But
Great point, yeah.
I also wonder whether it'd be interesting to combine these dynamic RSA and like the moments where you can say there's a shift in content, right? Like a correlation shift in the correlation structure that is the new content being represented with neural driving events, right? Because you can almost combine, right? Like, oh, is there an associated predictable, evoked response antecedent or preceding the representational change? And I guess I just immediately think, I'm like, okay, you could either do like an event window and just say like, oh, the 6 seconds before the change is when we should expect it, or you could do data driven, right? With like the iCraps, right? Or like some like ignition analysis, right? Like something that would like look for data driven events. Anyways, that'd be a cool way to combine the RSA with then evoke responses, which RSA is, of course, in some respect dealing with, but it's a little more
That's cool. And as we collect, we're starting to collect new data sets where we're going to be using this approach, Brandon is, but in the meantime, we do have this large data set like that. Just there's so much more.
ball pit. You can just jump in there. Yeah.
If I can just jump in and say, as somebody who's kind of in the trenches to Beck Todd on the project, I'm very thankful for all of this feedback we're hearing. We're going to be sure to integrate it. I mean, we've kind of almost subconsciously trying to get towards this direction that we're talking about now. Like even in our figures, we are instinctively leaning towards plotting correlations kind of at different time points and trying to show that in kind of a visual way. But we know that it's very coarse and it's kind of just a step in the direction we want to take. And it sounds like even if there's manifold ideas, that genuinely seems like the direction that we can take us further away from being constrained by just like these purely linear correlational approaches and looking at something that actually takes advantage of the mathematical tools that are given to us.
It's also, I think, an important, I made a comment in my talk about generative predictive models. I think this is kind of what we're talking about, right? Is the intersection of those two things. And you can stay in linear domain and get a lot of information from that. It's likely that a lot of the measures we're doing anyways are pretty linear as it is. fMRI, at least most of what we collect is still pretty linear until we get to sort of the sub-second TR stuff. But most of it are still doing like 1 or 2 second TRs, which is basically linear.
Another thing that pops to mind for me is like when if we're thinking about like manifolds, let's say, right? Like you have these like small movements around that state space, but they aren't constituting jumps, right, to other states that are still likely meaningful at a certain dimension of experience or psychology. Once again, it's not like we might I think put too much into these leaps, the whole brain manifold leaps in state space, whereas there's experientially perhaps relevant changes happening in that minutia of wandering in your local space. And it would be interesting to consider, I hate the idea of like going into an atlas and taking out a network and be like, all right, we're just putting that network in as a manifold. But like, you could use integration segregation measures to decide when it is appropriate and not in time series to segregate a network and then treat it as the manifold space. So look at like experientially smaller walks in content, right? That's what I'm thinking. I mean, how do you zoom in on
it's not just shifts between, but yes, within there's movement.
Yeah, and zooming in on those, and it would be finer grain phenomena.
Controversial today. Like, what if you zoom out and the network is your smallest unit of measurement? So now you have between networks kind of at that level. And so you might have the same state space for an entire experiment with multiple conditions, but then how those are connected is the difference. So
yeah, right. And that's why you'd want to, like, I guess before doing that, evaluate whether it's even with the integration segregation component, right? Whether it's valuable.
I think that's a very important point. You did it to a point that is very dangerous in some way.
Oh yeah.
So why it's dangerous is we're getting to a point in science right now that we're publishing differences between population of like 25 milliseconds, right? And there's tons of publication like that. And so, and it's significant because on the large population, you get that stuff, right? So those are very much variation that you're talking about there. And we tend to say, okay, it's significant, or that means it's something, but actually it's not really something. Like I give you an example, we work with scientists, with Randy And she showed like differences between bilingual and monolingual of in executive function of like less than 50 milliseconds. And that her whole career on that. Like for bilingual, like I'm nothing, those 50 milliseconds of gain that I have. Why are we talking about that in the literature? It's so important in bilingual literature. I think it's very important to get to zoom out a little bit and to try to really don't waste your time on those micro variations. That can be published, can be a difference, but really don't just too much about what is really the system and try to ask questions. Okay, we have one last question. I'm very sorry because
That's fine.
Just wanted to answer one of your questions and my opinion on one of your questions, which is whether TR is appropriate for measuring these trajectories in the brain. So I guess for me, the answer is that if you're actually interested in neural trajectories throughout the brain, the TR is not going to be working because obviously things happen too fast for having a measurement every 2 seconds. But you know, and I know you're using the bold signal anyway.
Correct.
That's not a measure of neural activity.
Yep.
That's a measure of blood oxygen level dependent changes in the brain. And that, this thing's so slow that a TR is awesome. You're at 2 seconds when this thing takes like 10 seconds to complete, you know, a cycle of doing anything. So
That's a good point. We're not going to be measuring MEG, EEG, speed of thought processes. We're measuring.
so yeah, I think TR is great for the data you have. It's not great if you're actually thinking you're going to be measuring neural trajectories throughout the brain.
Absolutely. Yeah, no, that's a really important thing to hold front and center because they need to forget.
I think that's an awesome comment to finish on the morning because with a lot of students there, one thing that I'm always kind of like surprised is when I talk to students that's computational neuroscience, they don't really understand the data. So please don't forget that. Like this is very important. You look at it from what am I looking at? I'm looking at bold signals. Right? And if you go to the detail of this science, you will see that what we measure actually in the papers is actually not the part of the signal that is the most related to the neural activity, right? So understanding your signal and your data is hugely important, and you should always go back to what are your data.
And if you ever need that reminder, then SFU folks come to UBC and hang out with Todd because you will.
I have same always reminder, and I think can tell you at each time they give a talk, I'm asking them, like, what are you talking about? What else is that? And we have to do that. So I agree with that. I don't think we can be enough. So thank you very much, Rebecca. Thank you so much for this inspiring talk.